Thanks very much, Austin, for getting us started. That's terrific. So let's move on to the next speaker then, Dr. Shengdar Tsai from Massachusetts General Hospital and Harvard University Med School. And he will talk to us about genome sequencing approaches and determining off-target effects of engineered nucleases. Okay, Dr. Tsai.
Okay. Thanks very much for that introduction. I'm an instructor in the lab of Keith Sheng, and I'm here to talk to you today about genome-wide off-target effects. So for quite some time, our lab has worked on targeted genome editing. And so simply, we think of this as the capability to introduce targeted alterations into any DNA sequence with high efficiency in any living cells or organisms. So more recently, we've become a little bit more picky about this definition. And so ideally, we'd like to do this also without making any other undesired changes in the genome. You may have heard a lot about CRISPR-Cas9 technology. Our lab has worked on a number of different engineered nucleases, including zinc finger nucleases and talons. But there's been a tremendous amount of interest in CRISPR-Cas technology. And the reason for this is that it really simplifies the process of genome engineering. And so to give you a really quick overview, the system is essentially a two-component system composed of Cas9 protein, which is complexed with a short guide RNA sequence. So CRISPR-Cas9 can recognize a 20 base pair target site adjacent to a NGG PAM or protospacer adjacent motif. And the basis of this target recognition is based on RNA-DNA complementarity. And that's really part of the elegant simplicity of the system that was originally developed by Jennifer Doudna's lab, and then demonstrated that it could work by both Jennifer's lab, George Church's lab, and Feng Zhang's lab. So the system can cleave its target site adjacent to this NGG PAM and introduce a targeted double-stranded break. So these nuclease-induced double-stranded breaks can then typically be repaired in mammalian systems, for example, by one of two competing DNA repair pathways, either the non-homologous and joining mediated pathway, where you can get variable length insertions or deletions, or the homology-directed repair pathway, where you can get precise gene correction. The targeted genome editing has become really quite simple with CRISPR. But one question is, what about off-target effects? So the recognition of these DNA target sites by simple RNA-DNA complementarity might have some limitations. This is something that we think about a great deal in the context of human therapeutics, where you might be treating populations of cells. But it's also very important to address for gene drive applications. So here's a figure that I've borrowed from Science, where you have this idea of the gene drive, and this is related to this mutagenic chain reaction paper that we've had recently. And so the concept here is that you're expressing a nuclease, so you're expressing constructs for both Cas9 and a guide RNA in the context of either cells or an organism. Some considerations are that the continuous long-term expression of these nucleases increases the opportunity for off-target mutagenesis. So you might have a nuclease that cleaves at low frequency initially, but over time can have high frequency mutagenesis. Another consideration is that unintended nuclease-induced mutagenesis can also be heritable, so they can essentially be carried along with the gene drive, and so that's something to think about. And so therefore, we'd really like to do the best possible job to identify all the off-target effects of engineered nucleases, even those that are low frequency. So the challenge is that you'd like a method that would ideally have three characteristics: that it would be genome-wide in scope, highly sensitive, and unbiased. One approach that you might immediately think of in terms of finding off-target effects of engineered nucleases might be whole genome sequencing. And the issue with this method is that it's not really practical to sequence large numbers of genomes. If you do standard whole genome sequencing, you might imagine that you might have a read coverage of maybe 50 or 100. But the limitation is that in that case, you really wouldn't be able to detect anything below 1%. You'll miss low frequency off-target effects, and you'll have poor sensitivity overall. Another approach that we and others have used is targeted sequencing of off-target sites. And so this is a two-step approach where initially first you identify the genomic sites that might potentially be off-target sites by computational prediction. This is based on the assumption that the off-target sites resemble the on-target site, and there's really a need to reduce the overall scope of the search by using these computational methods. You can imagine that as you go out to off-target sites that might have several mismatches, 5 or 6, you're starting to look at potentially thousands, tens of thousands of sites, or even more in the context of a whole genome. The next step would be to examine these sites by next-generation sequencing in cells that have been treated with the nuclease for evidence of mutation. One fundamental limitation here is that there's a background technical sequencing error rate. It's usually around 0.1% for next-generation sequencing technologies. And so that really sets a floor for the mutation frequencies that you can actually detect. But there's a more fundamental limitation of this approach, and I'd like to describe this as the streetlight effect. And so you have this example here where you have a cop coming up to a guy who's looking for his keys. And she says, is this where you lost them? And he says, no, I think it was 2 blocks over. And she says, then why are you looking for them here? And he says, well, because the light is so much better here. And it might seem kind of crazy to do something like this on its face, but it's actually a pretty good example of observational bias, and we think this is actually a pretty good illustration of the limitations of targeted sequencing. And so to extend that analogy, if you imagine the genome is a parking lot, then targeted deep sequencing is essentially like having a streetlight shining the light on very specific areas. But the limitation is that you'll never find things where you're not looking. So this is really what motivated the development of one of our approach called genome-wide unbiased identification of double-stranded breaks enabled by sequencing, or GuideSeq. So GuideSeq is based on the principle of efficient incorporation of genetic tags into nuclease-induced double-stranded breaks. So without going into the details, we optimize the efficient integration of an N-protected small oligoduplex tag into CRISPR-Cas9 double-stranded breaks. These tags are blunt and likely integrated by non-homologous end joining. So having captured these tags in living cells, then we can perform unbiased tag-specific amplification to recover potential sites of double-stranded breaks. We then sequence and map the amplified fragments. Here's a diagram illustrating what we might expect. So because the tag is blunt, it can integrate in either direction, and we use primers against both the forward and reverse strands of the tag, and we expect to see this characteristic bidirectional read pattern that would be a signature of a double-stranded break. So you expect to see reads that emanate from the double-stranded break to either the forward or reverse strand surrounding the breakpoint. So I'm showing you here an example at a gene we've studied before called EMX1. And on the x-axis here, I'm showing you the CRISPR-Cas9 target site. This is the protospacer sequence with the NGG protospacer adjacent motif on the right. So we expect the cut site to be where this red dashed line is, and we detect these GuideSeq reads precisely where we'd expect. You see a sharp peak at the predicted breakpoint with bidirectionally mapping reads indicated in different colors. So we expect the number of reads at each site to be proportional to the frequency of cutting and mutation. So now we've done this at a number of different, 10 different CRISPR-Cas9 RNA-guided nuclease target sites. We first looked at 4 sites with known off-targets that we've studied before. And we do this in 2 cell lines. And so this affords us the first genome-wide off-target profiles of 10 CRISPR-Cas RNA-guided nucleases. We're gratified to find that in the case of the 4 that we'd studied before, using this method, we were able to find all previously known off-targets. We're also able to find a wide variety of novel off-targets as well, so you can see a wide range of numbers there. Here's a chromosome diagram of the off-targets that we found. The on-target site is labeled in green, and then the off-target sites are in red. And so you can see that they're dispersed throughout the genome. And here's another couple of sites. You can see that there are wide ranges in the numbers of sites. This on the right here is a particularly promiscuous guide RNA, and so in that particular case, there's a high number of off-targets. And here's some data showing that there's pretty good correlations between experiments where we're conducting with both CRISPR-Cas nucleases and this genetic tag, as well as cases where we're treating cells with just the nuclease by itself, that we see pretty good correlations in the frequencies of guide-seq reads detected. Here's a plot of all the different target sites, target and off-target sites that we've seen in the study. I realize it's a little too small to see, so zoom in on that. So at the top, I have the intended CRISPR-Cas9 target site, along with the protospacer adjacent motif here on the right, NGG. And so these are, and then I've listed the off-target sites in order of frequency. The intended on-target site sequences at the top, and mismatches relative to the intended site are indicated with the colored boxes. So the intended target site cleavage frequency is indicated here, and it's marked with this black square. It's interesting to note that in, for example, these cases, there are actually off-target sites that are higher in frequency than the intended on-target site. The known off-target cleavage sites are indicated in diamonds. You can also see that these ones that were known before were ones with higher read counts, so you might have been detecting things that were easier to find earlier on. For CRISPR-Cas systems, up to 6 mismatches can be tolerated at these off-target sites. And non-canonical PAM sequences, normally we'd expect NGG, but so non-canonical PAM sequences can be tolerated as well. And so this is really setting up the challenge for computational prediction because there can be this high number of mismatches tolerated at the protospacer RNA-DNA interface. It means that you might have to computationally look for sites with up to 6 mismatches, and this would be potentially tens of thousands of sites in the genome. So here I'm showing you a comparison of guide-seq empirically detected off-target sites with computational predicted off-targets. In yellow, I'm showing you the set of computational off-targets. Blue is guide-seq, and pink is the intersection between the two. And these are 2 of the top computational prediction methods. I think the take-home message here is that these computational methods do not capture the majority of bona fide guide-seq detected off-target cleavage sites. And so that a lot is missed by computational prediction. This is not particularly surprising because a lot of these methods really only look out to 3 or 4 mismatches. And so anything with 5 or 6 is automatically missed. We also used a method called AMP, or Anchored Multiplex PCR, to identify potential large-scale genomic rearrangements that happen at these on-target and off-target double-stranded breaks. So essentially, we use a primer that allows us to sequence across these translocation junctions, and we look at the on-target site as well as all the off-target sites that we detected. Here's some sequencing data for some specific examples, and I'm showing you that we can detect, for example, at VEGF-A site 1, all potential reciprocal translocations, as well as large deletions and inversions. And so stepping back and taking a look at this on a genome-wide scale, here I'm showing you Circos plots. So these essentially have the chromosomes arranged in a circle, and genomic rearrangements or translocations are shown as arcs on the circle. And so the important thing to appreciate here is that we can not only see translocations between the on-target site, the off-target site, between different off-target sites, but also between off-target sites and background double-stranded breaks that might occur. So I've described to you GuideSeq, which is our method for identifying off-target double-stranded breaks. There are also other methods. Before GuideSeq, there was also this integration defective lentiviral capture method that was first developed by Richard Gabriel, Von Kalle's lab, as well as this high-throughput translocation sequencing method. So there are a number of different methods potentially for finding, genome-wide methods for finding off-target double-stranded breaks. So I think the take-home messages here are that unintended off-target cleavage can occur with CRISPR-Cas9 nucleases. This can happen with up to 6 mismatches, and also in sites with non-canonical PAMs. That in some cases, these off-target sites can be cleaved with equal or higher frequency than the on-target site, and so that's really important to consider in gene drive applications as well. So you can imagine that if you're driving this particular cassette in some species, that you might also be carrying these off-target mutations as well. That the computational methods fail to predict the majority of bona fide sites, and so using computational prediction is really inadequate for finding off-target effects of engineered nucleases. You can't simply take a look at, for example, the top 10 computational predicted sites, say that you haven't found anything, and conclude that this nuclease has singular genome-wide specificity. You really have to look at these carefully on a genome-wide scale. As I mentioned, that these off-target mutations could be transmitted as passenger mutations by gene drives, and so it's really important to think about gene drives in the context of the genome, that as you express an engineered nuclease, that it can act not only on its intended target, but also all of these potential off-target sites in the genome. And finally, that the risk of off-target cleavage should really be carefully considered and empirically assessed genome-wide using these empirical methods, not in silico methods, for potential gene drive applications. And with that, I'd like to acknowledge my mentor, Keith Zhang, the members of our lab, our collaborators, and our funding sources. Thank you.